Welcome back, ladies and gentlemen. We're going to continue wrapping up today your chapter 22 and really just looking at some of the evidence that exists uh, for supporting evolution, some of which Darwin has seen and some of which we've kind of discovered since his time. One of the biggest pieces of supporting evidence is this idea of homology, and homo is that prefix that means same. So we see similarities between organisms and we point to that as evidence for evolution because those similarities must have come from their common ancestry. And we often end up mapping these homologies in things like these evolutionary trees, which are simply these hypotheses about the relationships among different groups of organisms. The idea here is the more homologies that are similar between species, the more traits that they share in common, the more recent their divergence from each other, and the fewer things they share, the further back in evolutionary history we have to go in order to find their common ancestor. So if I look at something like the hawks and the ostrich, they share all of these traits in common, and so their split from each other in terms of an evolutionary branch is much more recent. That being said, I look at the hawks versus mammals, and they only share a few things in common. So I've got to go back even further in evolutionary history before I find the common ancestor that links them together. Now, when we talk about homologies, we're talking about similarities, and these similarities can be anatomical, which is often what we tend to focus on as underclassmen, are physical characteristics. And that's what you're seeing here in this picture. Do they share feathers? Do they have an amniotic egg? That kind of thing. But we can also look more recently at molecular data, DNA, amino acid sequences, that sort of thing that they can potentially share in common. And that gives us yet another piece of evidence to look at that isn't physical, and we start digging into their genetic code. When we talk about Darwin, he would have been looking at all the anatomical similarities because he didn't know DNA existed. So that DNA homology, that protein homology, that molecular data is much more recent as we've been able to sequence that kind of information. So when we talk about anatomical homologies, we're talking about homologous structures, structures that are in different species, but they share a common ancestry. And this diagram here is probably the most famous and most widely used example of homologous structures. And we're looking at the forelimb bone structures in these different organisms. Now, if you look, when we talk about homologous structures, we're not talking about structures that organisms necessarily use in the same way or are even necessarily structured identically. That being said, we can sit here and you can trace the different colors for the different bone structures. And although the shapes and lengths and widths, if you will, of those different bones may be slightly different from a human to a cat to a whale to a bat, they're all there. And the other piece to this is as we've started digging into how they develop from embryo to adult, the pathway in which they develop is also similar. So when we talk about homologous structures, it's all often bone, muscle, nerve orientation, that sort of thing that we're looking at. And it all ties back to also the developmental pathway from fetus to adult, from embryo to adult, how these structures come into place. Here's your idea of molecular homologies. This one happens to be looking at amino acids. And if you remember your sort of central dogma, right, your DNA codes for mRNA, which then gives us the code to build amino acids, and amino acids are the building blocks for proteins. So all of this is very tightly connected, which is why we can use DNA sequences, we can use mRNA sequences, we can use amino acid sequences because it all ultimately ties back to your genetic code. And remember that for evolution to function, we need to be able to pass on that genetic code to the offspring. And ultimately, the protein that gets produced is the phenotype that we see, and the phenotype is then what natural selection is working on. So it's all very tightly connected. This particular molecular homology is looking at the amino acids for human hemoglobin. and we get these percentages of comparison. So if I look at human to human, hopefully that makes sense that we should have 100% identical amino acid sequence because it's human. That being said, if I compare human hemoglobin's amino acid structure to the hemoglobin we find in a rhesus monkey, about 95% of the amino acids are identical. Down to the mouse, 87% of the amino acids in order are identical, so on and so forth. And so we can use this to suggest that that rhesus monkey shares a common ancestor with humans much more recently in evolutionary history. I have to go back considerably further in time in order to find a common ancestor from a human to a lamprey, and that's why there's been so much change over time, and we only share 14% of that structure in common. Now, something I wanted to point out while we're talking about structure and homology is its opposite. So, when we talk about homologous structures, we're talking about structures that develop similarly over that organism's lifetime. Not necessarily that they look 100% identical, and not that they necessarily are utilized in the same fashion from one species to the next. It's a developmental pathway that's the same, as opposed to an analogous structure, where analogous structures aren't true evolution. It's convergent evolution as opposed to divergent evolution. So when we're looking at things like this or like this is divergent evolution. We start from a common ancestor and things change over time as we branch out into these different species. When we're talking about convergent evolution, we're coming together on a similar structure for a similar function, not because they share a common ancestor, but because they fit similar niches in their environment. So a sugar glider and a flying squirrel don't necessarily have common ancestry. Recently speaking, I'd have to go really, really, really deep into the evolutionary tree in order to find any sort of common ancestry between them. So when we're looking at the structure of their sort of flaps of skin and the way that they sort of glide from tree to tree, isn't because they share common ancestry, but because the sugar glider in Australia and the flying squirrel in North America fit very similar niches in their ecosystems. And so they happen to have converged on very similar structures because of the way that species survives in their environment. So convergent evolution, analogous structures, not true evolution, but often mistakenly cited as evidence. So just make sure you're careful with that. Here's comparative embryology. This takes the homologies to another level, and now we can start looking at developmental pieces in embryos and how that changes from one species to the next as that structure develops from embryo to adult. So if I were to have taken these labels out of here and shown you this human embryo and this chicken embryo, it may have been relatively difficult to know which was which. But if we look, the structures are the same in the embryos. And then as they develop into a baby, into an adult, those structures change and perform different functions. So something like your bones of your inner ear, all right, are actually gill slits as embryos in humans, while fish have those same things. They ultimately stay as gills in that fish species, but in us, those gill slits become the bones of our inner ear. So it's molecular timing, it's turning things on and off at different times that have changed the way that that structure ends up developing. And that's a result of changes in genetic timing in development, and that's some stuff that we'll touch on a little bit more this year as well. Vestigial organs, all right, also another piece that we tend to point to as support for evolution. Now, when we talk about vestigial structures or vestigial organs, these guys are pieces that we see in current organisms that presumably had some sort of function in our common ancestry, but they no longer really seem to do anything for us now. be careful with this idea of vestigial organs. A lot of kids want to throw in that use versus disuse piece. Um, so this evidence often ends up being misconstrued with Lamarckian evolution. Be careful with that. What we end up truly seeing with vestigial organs is that they are historical remnants that probably serve some sort of function in those ancestral organisms. For whatever reason, they lost that ability, probably some sort of genetic mutation caused them to sort of get shrunk, caused them to lose that function in their ancestors, but they still hang around. So, why is that? Well, when we talk about vestigial organs, these are pieces that, generally speaking, don't really cause any harm to the current living species, but they're not beneficial either. So, they kind of just hang out. Natural selection doesn't work on them, so a genetic mutation probably arose somewhere along the way to shorten it, to lose its function, and it wasn't detrimental to the species at that point in its evolutionary history. And so we continue to see this stuff kind of hang out. Um, so whales, for example, still have a pelvis, and they'll probably never lose it completely because it's not harmful to them now. It's not beneficial to them either, and so it just kind of stays there. Our tailbone, our appendix, generally speaking, most people can live their entire lives without their appendix bursting. So these things will just kind of hang out. Maybe we get a mutation that sort of starts to pervade in the population and it disappears, and maybe not. There's really no way to understand what's going to happen to these particular structures because they're not necessarily harmful or beneficial, and so natural selection doesn't really do anything with them. Biogeography, this is an interesting one where we can start tying to the understanding of things like plate tectonics and the fact that land masses all existed as this big Pangea at one point, and so species were able to travel around to different places, and as those plates separated, that's how things got geographically isolated from one population to the next. They can no longer get to each other. Kind of like the idea of Darwin suggesting the finches looking similar to the birds on the mainland of South America, and then those islands branched off from each other, and that's where you get that diversity of different finch species from land to land, from island to island. So what we can see here is we often find similarly developed organisms here on the coast of South America as we do in Africa because look at them, they fit like puzzle pieces, more or less. And so presumably these were in fact once connected, and the theory of plate tectonics helps to support that idea. Fossil record is another wonderful example. Again, go back to Darwin seeing the fossil of the giant sloth and then seeing the real sloths in jungles of South America, the fossils at the top of the Andes Mountains of aquatic organisms, all this kind of stuff. So remember, your fossil record is that successive order of organisms. Deeper you go, assuming those rock layers haven't moved, the deeper you go, the older the organisms are back in time, and then we move forward as we get closer to the surface. So those fossil records are a great way to see that successive form. If we can in fact find transitional fossils, it's even better. So being able to see something like those changes. So here's our current living whales, but we also have found transitional fossils where they used to have legs. So kind of an interesting connection there as well. The other interesting thing that we can use to cite evidence for evolution is the fact that at least we've got living examples. There are examples that we can watch, we have studied. And so natural selection in this particular example is because of an introduced plant species. So we look at these little bugs here, all right, and their beak length based on their ability to tap into their food source. And it has changed once we had an introduced species. So take a look at this soapberry bug example, and it's kind of interesting. So they use their beaks to feed on the seeds within the fruits, which is what you're seeing here, right? And so their little beak is is out here. And so they'll use those beaks to feed on the fruits. When you looked at the species that would typically be found in southern Florida, they tended to feed on these balloon vines that had these big, large fruits. And as a result of being able to tap that food source, that meant that evolution had pushed them to have these sort of longer beaks to be able to reach through those bigger fruits. You went more towards central Florida, and these guys fed on smaller fruits and thus didn't need to have that long beak. The shorter beak was probably beneficial in the fact that they could get their food and probably wasn't as energy intensive to produce as a population. And so there was this correlation between fruit size and beak size. And Florida wasn't the only place that we saw this. We saw this in other places like Louisiana, Oklahoma. So in all cases, those beak sizes evolved in the populations that fed on the fruits that were naturally found. Interesting thing, introducing a different plant, a different fruit than the one that they were typically natively attached to, and we see this shift. So if we're looking at this, we get this introduced species. So here's where they typically were existing, and now they end up getting the average size, if you will, gets shifted as they get introduced to different size fruits. So for the guys who were typically in southern Florida with these bigger fruits, if the smaller ones the smaller fruit plants got introduced, their beaks would actually get shorter, and vice versa. So we were able to actually trace this evolution because these fruits got introduced into these native populations over 35 years, and we were able to watch how their beak shape actually evolved and changed in its length based on the availability of these more introduced fruits that weren't naturally there. So it's just kind of an interesting case study in natural selection that we were able to watch within a relatively short period of time. This is another one, and we're watching this happen more and more and more, is the evolution of drug resistance in bacteria. Evolution in pesticide resistance is another really great example that AP loves to take into account. So first off, let's take a step back here, right? So when we're talking about bacteria, these are typically bacteria we study that cause some sort of a disease or just happen to live naturally within our microbiome, so on our bodies, in our bodies, that sort of thing. Along came antibiotics, right? Drugs that end up killing off these pathogen-causing bacteria. And the more we use the drugs, the stronger the selective pressure. And what do I mean by that? The more often we use them, the quicker the bacteria that naturally had some resistance in their population become the majority of their population. And so it ends up causing this to evolve rapidly. Now, we typically talk about antibiotic resistance, but the same principle probably is going to apply to things like antivirals. The more that we end up using those, we're going to take those susceptible portions of the population, right? The bacteria or the viruses that are going to be killed off by the drug, leaving behind the ones that are more resistant to it. And so Staphylococcus aureus is a very common one that's been studied. It's commonly found on your skin. Generally, most people don't have a huge issue with it, but you get some sort of puncture wound or something like that, and occasionally you end up with a staph infection. It became a huge problem when we started finding out that these staph infections weren't being cleared by the antibiotics we were using. And that's where things like the methicillin-resistant Staphylococcus aureus or MRSA comes up. And methicillin is not the only antibiotic that we find resistant bacteria to. Penicillin was the first antibiotic, and this was supposed to be the wonder drug. It was going to cure everything, and so doctors were prescribing it literally for everything. And within the first 2 years of being widely used, we were already seeing huge populations of bacteria being resistant to it. Methicillin came about in 1961, and within 2 years after its use, it also started having huge populations of resistance, huge pockets of resistance to the methicillin. And this continues to happen. So when we look at natural selection, remember that evolution is working on a population. So any population is going to have variation within it. If we're talking about bacteria and drugs, well, these antibiotics are derivatives of naturally produced compounds, usually from plants. So it's stuff that they've always encountered in their populations in nature. So you've got one end of the spectrum that is incredibly resistant. You've got the other end of the spectrum that's incredibly susceptible. Well, the susceptible population, or the more susceptible, I should say, end of the population, is going to get killed off by the antibiotic, leaving behind the resistant piece. So it's incredibly important when we talk about using antibiotics in an effective and safe manner. So its dosage, its length of use to ensure that we're clearing your infection and we're not leaving behind those resistant populations. We leave them behind, that's when they spread, and that's where we get these problems. That being said, even if we use the course of antibiotics correctly all the time, eventually we've got to up the dose, we've got to use it longer in order to clear the infection, because this is a great example of evolution. The more we use it, the more we continue to select for the resistant population, and the more that becomes the dominant trait in that population. So methicillin specifically works by inhibiting a protein used by bacteria to create their cell walls. So what ended up happening is we discovered that there was a variation in the different types of proteins that were being used. When we look at bacteria that were being killed off by the methicillin, they were in fact the portion of the population that this protein that was being inhibited functioned on. It did its job, it destroyed the bacteria's ability to build their cell walls, so they died off. But within that same population are these MRSA strains, these bacteria that use a different protein that methicillin can't stop its production. And so nature was selecting for that different protein, and all of a sudden the bacteria that we were seeing in colonies were producing this different protein. That means methicillin didn't work. And so interesting thing with the MRSA and a lot of drug-resistant bacteria is that it's often not just resistant to a single antibiotic. They're often strains that are now resistant to many antibiotics. So we're starting to need to find other ways in order to tackle bacterial infections, because we're running out of antibiotics that are effective. There are some things out there on the sort of research field, things like phage therapy, going after them with viruses instead of antibiotics. But a lot of this is still being studied. It's not necessarily being mass used. So it'll be interesting to see how we kind of take care of this problem, because it's going to become a big one if we don't.